DNA is important for us as the storer of genetic information, and that genetic information, as it's stored, is then used to be translated and used in the form of proteins. So basically, we usually say that DNA is stored information, and then it's used to facilitate and to direct the synthesis of specific proteins. So in order to go from DNA to proteins, we actually have to use an intermediate step as well, which is going to be the formation of RNA. Um, so basically the general flow of genetic information, and this is actually on a subsequent slide, but I'm going to draw it on here as we talk about it. It's going to be DNA goes to RNA, which then goes to protein. And more specifically, whenever we talk about this RNA, we're going to call this one messenger RNA or mRNA. So there's actually a number of different types of RNA out there, but this messenger RNA is the one that directly leads to the formation of protein. In addition, right, our cells in the body frequently divide. And whenever those cells divide, it's important that they get a copy of that DNA. So there's also a process where DNA can make more DNA, and that's this first one we're looking at here on the second bullet point where it talks about replication. So replication is a process where DNA makes a copy of itself. So this occurs when there's cell division going on so that the DNA basically makes another copy of the DNA. Now transcription is going to be the process from going from DNA to RNA. So transcription is going to be this process right here where you go from DNA to the messenger RNA. So this is making RNA from a DNA molecule. And that's going to be the genetic information is now going to be temporarily stored on this messenger RNA molecule until the messenger RNA molecule can be converted into protein through a process called translation. So translation is when you're going to go from messenger RNA to protein. So translation is basically translating. It's kind of like if you were to translate Spanish to English, right? You're taking it from one language to another. So this translation is translating from messenger RNA, which is going to be a language of nucleotides, into protein, which is going to be a language of amino acids. Just to finish off the slide here, right? We can also say that replication is this process here where the DNA makes some more DNA. Okay, so now that we've looked at all of these, let's go to the next slide, which kind of shows the same thing I just was talking about with a slightly different twist to it. So the replication part is where we're going to start. So looking over here at replication, it has a little bit more detail with the words from parent DNA to daughter DNA. So the idea here is that the parent DNA is what you start with. After replication, you end up with a daughter DNA, and specifically, you start with one strand of the parent, so one strand of parent, and you end up with two strands of the daughter DNA. So we're going to go through and look at replication, transcription, and translation kind of one at a time, but the first thing we want to do here is look at replication. That's going to be our first step. So DNA replication is the formation of two new DNA molecules. Again, these are referred to as daughter strands. If you look at the slide here, it says daughter DNA and daughter DNA. Both of those daughter strands are going to be identical to the original DNA. And if you follow the color scheme here, so in the parent DNA, you have this blue strand. And one strand is going to be ATTCGA, right? And let's just let me go in and label this just for clarity's sake. So let's call that the 5' end and that the 3' end. And that means the other strand, if you remember, DNA is always anti-parallel. So they're going to be, one's going to be from top to bottom 5 to 3. The other one's going to be bottom to top 5 to 3. A base pairs with T, C's base pairs with G. So in order for this replication to occur, you're going to take these two strands here and you're going to separate them. And as they're separated, additional strands get made, and that's how you get the two daughter strands. So if you notice in your daughter strands, you have one of the original copies still in blue here, right, from the original parent, And then you have the one in red is going to be a newly synthesized strand that is going to be made from scratch. So your daughter DNA has one of the strands originally from the parent, and the other one is going to be a new strand, which is synthesized kind of from scratch. All right, so let's look at how this happens. The idea here, and I'm going to start down here on this third bullet point where it says replication occurs in only one direction on the template strand from the 3' to the 5' end. So I'm going to try to draw this out a little bit down here on the bottom. So if we take a DNA strand, so 5', and let's just make a really short one like this. So just 6 nucleotides. So just kind of imagine though, remember a lot of DNA strands are like millions of nucleotides because something that I'm going to add here is going to be important in that here in just a minute. All right, so we would have a double-stranded molecule that looks like this, right, anti-parallel. And that's going to be the original parent strand. So DNA, again, imagine it's a really long molecule. It's going to start unwinding. It can only unwind from one end to the other. So if I go back to the previous slide real quick, imagine if we were going to start replication up here, right? So the top part of this DNA is going to unwind, but the bottom part's going to stay together, right? So imagine the top part being unwound here. And what's going to happen is whenever it unwinds, is you're going to basically separate these two strands. So I'm going to go in here and I'm going to actually erase what I just put. And now I'm going to redraw it, the same thing. I'm just going to draw it further down because the idea here is that we have kind of separated these strands a little bit. All right, so those are going to be our 2 strands, now slightly separated. Replication only occurs from the 3' to the 5' end. This means that whenever we start adding the new molecule, we are going to specifically add it so that the new strand grows in a 5' to 3' direction. So we're going to, this is actually a true statement, right? The replication occurs from the 3' to the 5' end of the template. Another way to say this, and this is actually how my brain prefers to think about it most of the time, is new strands are synthesized from 5' to 3'. Okay, so in other words, our new strand as it's going to be made is going to start from the 5' end and it's going to grow to the 3' end. So what does this look like? So the easier one is going to be on the bottom here. We're going to draw a new strand. So the new strand down here is going to be in red. All right, and it's going to build from the 5' to the 3'. So we're just going to add an A here because up here on the second bullet point, A must pair with T, G with C. So the enzyme that does this work recognizes that there's a T down here, and it says, okay, I need to add an A here. There's an A here, so it needs to add a T. There's another A, it has to add a T. There's a T, it has to add an A. G goes to C. C goes to G, and we end up on the 3' end. So this down here would be one of your daughter strands, okay? Now, the tricky part is on the other one. And the reason the other one is trickier, and I'm going to draw this one in blue, is because of this idea that the new strands can only be synthesized in a 5' to 3' direction. So that means that as this thing unwinds, right, ideally we would like to say, okay, there's going to be an A there and we're going to put a T here. And there's going to be a T there and we're going to put an A right beneath it. The problem here is we would be building from a 3' to a 5' direction, and we can't do that. So what happens with your DNA is it becomes partially unwound. So imagine that half of this molecule was unwound. So the A and the T were unwound, but the ACG was still all kind of together in the double helix. So what happens is it gets partially unwound, and it actually gets synthesized in this direction, A, T, because right, that would be growing in a 5' to 3' direction, right? It's going to grow this way. So first the A, then this A, and then the T. So it's going to grow in this way. And then more of it gets unwound, and then you're going to have a C, a G, and a T. And it goes this way. So the idea here is on one strand, you're going to basically continue making it in, we call these small fragments, right? And they have a fancy name called Okazaki fragments based on the scientists who discovered them. But the idea is that you're always going to be building in these small fragments, and then the small fragments are eventually linked together at the end. And this is referred to as the lagging strand. So the lagging strand is the one that's built into small pieces. Now, if we look at the other one, this one down here that was much more direct, where we just took the 5' end and went directly to the 3' end, this one we're going to refer to as the leading strand. So whenever you have your parent DNA molecule and you form the two daughter strands, you end up with two identical pieces, but they get there in two different ways. Right? The lagging strand and the leading strand are both made at the same time, but they're made slightly differently based on the principle that these new strands are always made in a 5' to 3' direction. So that directionality of the DNA is important for directing how this works. All right, so this here is just shows you some of the details that go into the synthesis of, this one says synthesis of the lagging strand, but the leading strand would have the same components. The DNA polymerase, so DNA polymerase is an enzyme. Remember, enzymes end in ase. So DNA polymerase is going to bind to and be important in basically polymerizing and making new DNA. Nucleoside triphosphate, so these are your basically the nucleic acids or the bases that get added in one at a time as the complement. All right, so if the green one was a G, then this one would be a C, right? And so these are the kind of some of the components that are important. So all of those nucleotides that we learned about, right, the A's, C, G's, and T's are all important here in order to be able to make and replicate your DNA.